Hello guys, this is Tushar Biswas and welcome to the Study Companion. Today we are going to learn about DNA in a more detailed format because this is the second lecture, second video on DNA, the introduction of DNA. Like what is DNA? Yeah, in the previous video we had seen that DNA, deoxyribonucleic acid in its the basic term. In the basic term we had seen it in. Now is the time we see that in a more detailed fashion. How is it packed? What are the distances between two base pairs and everything? What are the distances between two base pairs? Come on, man, that's mind-boggling stuff. And you're going to enjoy the video. I hope so. It's pretty informative. Please keep your books and copies ready wherever you like to note it down. If you note it down in your mind, of course, well and good. Go ahead and keep watching the Study Companion. Okay, so without any further ado, let's begin. One more thing, guys, before we start. Guys, after this lecture is complete, another video, this video is uploaded. Okay, after that we'll come across another video that will be a detailed video that's for people who would like to watch it. Okay, it's not mandatory for you to watch. If you want to have detailed information on how does an adenine, guanine, cytosine, thymine, and uracil looks in their own molecular formats, in their what are their conformations, I will bring a video on that, and that too with a sugar. I'll attach them in between themselves using those molecular pairs, using those molecules. I'll show you how the molecular pairing occurs. Okay, how the molecular pairing occurs. I will show you that in the next video. That will be a shorter video, but that will be informative. That will be actually in a detailed format. It's not required for your exam. It's for you to know. If you want, if you have the thirst for knowledge, hunger for knowledge, it's time to quench your thirst as well as nullify your hunger. So okay, keep watching and stay tuned. Okay, let us start without any further ado. First, the first thing we are going to speak of is the characteristics of DNA. Yesterday or in the previous video, we learned that DNA, like how does it look, why is it known as deoxy and stuff. Today we'll get to know why is it known as an acid. Why is an acid? That will come a bit later on in the video. So keep watching, and we are going to learn about things like how is the DNA packaged, because DNA is pretty long, and then the various characteristics of the DNA. So without any further ado, I would like to start the topic. Okay, is everyone with me over here? Let's focus. The characteristics of a DNA. Characteristics of a DNA. Now, what is the characteristics of the what are the characteristics rather? What are the characteristics of the DNA? Of course, there are five characteristics of the DNA. The five characteristics of a DNA, and I'll be producing you with the write-up in the picture to come over here. So please do copy the picture. Copy the picture down in your copies. You will get that in your NCRT books. You will get that in your own books you follow. But of course, you can, if you want, you can always copy this one down, which I'll be providing you in the next part. Okay. So, okay, guys. So I hope everyone has copied it down. Now, focusing on each and every part. Okay. It will go a bit random because everything I show over here will go hand in hand. So it might get a random in the writing column. It might get a bit more random, but each and everything will be explained. So don't worry. Number one. Number one, DNA. The the DNA strands were anti-parallel. If you remember in the last video, I had drawn those DNA structure. I had drawn the detailed DNA structure, though I hadn't focused on those nitrogenous bases which were just kept as in blocks. I didn't draw them, rather I didn't draw them in the actual format. So please focus over here. I will be clearing all your doubts if there is any. Of course, if there is none, then you people are studying. Still, if you want to follow, you can always follow this. You can always follow the topics. So number one is the DNA chain is anti-parallel. If you remember from my previous video where we had drawn it, if you haven't, the I button is over here. Go ahead and check it out. It's towards the end of the video. Okay, it's towards the end. Please check it out. If you haven't, if you want to know, so please check it out. So we know that DNA strands, they are anti-parallel in nature. Anti-parallel lines. These are parallel lines. So are these lines parallel to each other. That is, they have one direction. They're flowing in one good direction. One direction. When if one line goes like this and another one goes the opposite of it, this is known as anti-parallel. This is known as anti-parallel. These are parallels. These are parallel. See, if this is the direction, the one going in this direction is parallel to the direction, this one is anti-parallel to this one. So this one is parallel to this direction. This is the direction. In physics, this is the one which differentiates in between a unit in between scalars and vectors. This direction is the one which causes the rift between scalars and vectors. I'm not focusing on those parts, of course. This is the direction. For example, if this is my positive direction, this is my negative direction, this is my, oh yeah, let me have a graph. I'm not going to in-depth physics. This is a small part of physics. If this is positive direction, this has to be negative direction. We know it through vectors. Okay, we know it through vectors. If we take displacement over here and displacement over here, this one would be negative, this one would be positive. So would be the velocity, so would be the acceleration. Okay, any line which is parallel, for example, if a car is going in this line, the positive displacement, it's going in this line, it's going in this line. Okay, this line, the direction is the direction which we have stipulated for it is in positive because it's going from here to here, here to here, here to here. But another car is coming from here to here, but according to our frame of reference, our stipulation of the graph on this road, this car which is moving from here till here, it's coming from positive to negative direction. So its displacement is negative. For example, this is car A, this is car B. A's displacement would be positive because A is moving in the positive x direction, x-axis, positive x-axis, and B's displacement will be negative because B is moving in the negative x-axis. Clear? So this one is anti-parallel. Because why anti-parallel? If this is 5 prime and this is 3 prime, remember this 5 prime 3 prime, phosphate up over here, phosphate down over here. This always has to be 3 prime 5 prime. If you haven't seen my previous video, please go and check the previous video before you focus over here. And if you know it, you can as well as continue. You can as well continue. Okay, if you know why is it anti-parallel, of course you can as well continue. If you haven't, I have a detailed diagram on why did this happen. I have a detailed diagram on it. You can always go into my previous video and you can check it out. It's towards the end of the video. Please check it out. The link is in the I button already. You might as well check it out. Okay, coming to the second part. Coming to another part. This might not go in sync with the synchronization. I won't be using short terms. Synchronizations of what do you say of the one I had provided you with, but it's same information, just the numbers are up and down over here. Okay. Secondly, Secondly, the DNA codons. What are the codons? Which are the codons? Like what are the codons? What are the codons? Codons are the nitrogenous bases, N2 bases or NB, whatever you like to say. Whatever you like to say, you can say N2 bases, NB, whatever you like, codons. For example, you have adenine, guanine, cytosine, thymine, uracil. Hmm, these are the nitrogenous bases. Now these bond with each other. Remember the last video? A bonds with T. Is it double bond? Is it double bond? That's my question to you. Is it double bond? And is it triple bond? Now tell me in the comment section. Yes or no? If your answer is yes, my dear, you're on the right track. If it is, if by any chance it's no, you need to work on your skills. You need to work on your studies. Okay. So I would again suggest the previous video. It shouldn't be no after all. Still, it might as well happen for some students. It's okay. Sometimes it's good to make mistakes. It's actually good to make mistakes. We learn from it. The thing we learn from our mistakes, that thing we never forget. Okay. We never forget the thing we learn from our mistakes. We never forget the thing we learned. See, this is the whole thing. DNA strand, 5 prime, 3 prime, 3 prime, 5 prime. Remember anti-parallel. This is going from 5 to 3, then 5 to 3 again. It moves from 5 to 3. Remember this, DNA strand always moves from 5 prime to 3 prime. You will be needing this when we will be doing biotech and DNA replication, when we'll be doing central dogma. Okay, when we'll focus on central dogma in each and every part of central dogma. Okay, including replication, transcription, translation, we'll be focusing on those parts. Each and every minute details will be done. Okay, minute details as in whatever is required for class 12, both biotech as well as biology. Whatever is required, that thing will only be done. And for of course your NEET exams, nothing more than that would come in your exam. If you want to know more than that, of course the comment section is all yours. Show your love over there, comment it down, and I will make a dedicated video on it. So these are my DNA strands: 5 prime, 3 prime, 3 prime, 5 prime. Right, right, right. These are the codons. These are the codons. Nitrogenous bases sticking inside the DNA. Of course, these are straight strand, not the curved ones, which was suggested by Watson and Crick, Mr. Watson and Mr. Crick. Okay, this was done. And there is adenine, guanine, guanine, cytosine, adenine, thymine. So, so one more set of information, one more set of codons. A pairs with C. No, no. A pairs with T. G pairs with C. A again pairs with U. So we know that when there is a strand, there is one strand, and one of the strand has its own codons. We can always find out the codons present in the other strand. We can always find out the codons present in the other strand, because we know that these codons are opposite to each other. These codons are opposite to each other, of course. Hence, this A will always bind to T, G will always bind to C, G will always bind to C, C will always bind to G, A with T, T with A. See, two bonds, three bonds, three bonds, three bonds, two bonds, two bonds. Right? Correct? Correct me if I'm wrong. Anywhere. I don't think so. Let me check it out once more. Yeah, it's correct. This is the whole thing. This is the whole scenario now. The codons are opposite to each other. Okay, nitrogenous bases are opposite to each other and situated in between the sugar phosphate backbone. If you remember the term sugar phosphate backbone. Okay, sugar phosphate backbone. And for people who are having any kind of issues with me speaking in English, explaining in English, if you people want any other language to be spoken to you, to be like able to understand, of course comment it down. I can explain in Hindi, I can explain in Bengali. হিন্দি होता है यहाँ पे, I can always explain in Hindi, এখানে বাংলায়ও বলতে পারি আমি, no problem out there. Even if there is one person asking for this video to be again, not only this video, any of the videos of mine, if that person has asked, has requested me, not request, you can always ask, request, whatever you like to term it. If you people have asked, even if there is one person who is asking for it, I'll make a dedicated video, and that's my promise for you people. Of course, feel free to comment it down. The comments are all yours. Okay, because no student should go unheard. Everyone has the right to knowledge. Codons. Okay, this is clear. Erasing it out. Erasing it out. Now, now we know two things, and the third and one of the most important stuff which we are going to discuss right now is that we have seen DNA like this always, right? We have seen DNA like this. Then why am I drawing it like this? Have I gone mad? No, no one is mad, my dear. No one is mad. Okay, it's for your explanation. It's for your explanation that I'm drawing it like this. It's basically like this. Okay, I'm just straightening out the strands so that students can understand it better. Okay, please don't consider me mad for this. Thank you. Yeah, I'm a bit mad. It's okay. People tell me every now and then. Uh, but what to do? Okay, now let's focus on DNA. I'll be using two colors to explain the strands, to define the strands properly. Let this one be the 5' to 3' strand, and this one, the anti-parallel one be 3' to 5'. Okay, the strands are always inverted in the right-handed fashion. Inverted in the right-handed fashion. Now, how do you understand which one is right-handed? How do you understand which one is right-handed? Of course, that's a very basic question. How do you understand whether it's right-handed? Take up your right hand. Pick up your right hand. This is your right hand. For me, this is my right. For this, and I guess this is your left. Pick up your right hand. Pick up your right hand and your left hand, and then start. And then first force your right hand in front and your left hand at the back. Think there is a connection in between them, which does not let them come together. But if this one moves, this one has to move. See, this is what right-handed fashion means. Means the right hand goes in for the first turn. First turn, it won't go back. It would go in front. It will push in front. If it's like this is the joining. Over here, this will push in front. Hence, the left hand will go at the back. Right hand proceeds, left hand recedes. This is right-handed fashion. Right hand precedes, left hand recedes. Okay, guys, have I made myself clear? If I haven't, please let me know in the comment section. Okay, so now guys, consider this one is your right side. This one is your right side. Okay, right side. And then and this one is your left side. This one is your right side. This one is your left side. Now this right side, as I said, right side comes in front and the left side goes down. Left side recedes, right side precedes. What will happen if we take one single turn? Left, down, right, up, and like this. So guys, this is your right hand side, this is your left hand side. I have purposely written it for the students to focus more on the structures rather than actions. Okay, and not structures, I will do some actions of course, but if by any chance mirroring effect, the mirroring effect occurs and you people feel that somewhere or the other I'm showing the left side is coming is preceding and the right side is receding against the law, which goes against the law. There will be a huge confusion. Okay, and the mind will be filled with doubts, which I don't want. Okay, and hence I've written this is the right side, this is the left side. Of course, now this one takes a one complete turn. Right side precedes and the left side recedes, and boom, done. First, since the left side is receding, it will be somewhere like this, and the right side would always overlap it. This part won't be there because that's one single turn I'm talking about. Okay, this is one single turn. See how the right side has come in front and the left side has receded back. Right, left side has receded back. Look over here, the blue one was drawn first and the black one is overlapping the blue. Since we can always say that the left side has gone back and the right side has come in front. Of course, you can use this as a reference, right side, left side. Now for for the example, consider these two. This is my right side, this is my left side. This black is my right side, blue is my left side. These are like this, and these two markers are my nucleotides. This is my first nucleotide, first connection, and this is my second connection. This is my second connection. Okay, this is my first. Is the same thing would be over here, right side, left side. Okay, now the right one comes in front. I'll show it at a different perspective. I'll show it at a different perspective. Okay, let's come over here. Let's come over here. I'm shifting my place. This will be a better option. The right one, black, left is blue. Right one now comes in front while the blue recedes, while the left recedes. See over here, boom. And now can you any way, like can you by any way, any means, can you see anything except these two strands? Anything except these two strands, can you see? Can you see the nucleotide? I guess no one would be able to see that. From your point of view, from your angle, can you see the nucleotides? I don't think so, because you won't be. See, the nucleotides have gone invisible. But since it has gone invisible, you cannot negate the presence. The presence is always there. Presence is over here. The presence is over here. And then this again turns one single turn from here till here is one single turn. From here it's turning. Suddenly the nucleotides vanish because of our point of reference, point of view. Only the strands can be visible. And then see, black is overlapping blue over here. Same thing. And then again it's coming back to normal, like this one. Again back to normal. With the nucleotides visible. So the nucleotides are present all over in one turn. In one turn, for your information. No, not information. That's important actually. It's present over there. In one turn. This is one single turn. One single turn of what? Of the DNA. One single turn. This is one single turn of the DNA. And one single turn of the DNA. And it is also known as pitch. It's also known as the pitch of the DNA. Consists, first it's known as the pitch, secondly it consists of 10 base pairs one each and every pitch. Okay, so guys, now excluding the convention which we are following, let's draw a random DNA part. Okay, random DNA part. So how many pitch are present over here? Can you tell me how many pitch are present over here? Okay, now it will make it more clear. How many pitch are present over here? Tell me. Four or two or one or three? Which one? Which one? How many? How many? How many? How many? How many? If your answer is four, then my dear, you're absolutely wrong. If your answer is one or three, you're again wrong. The actual answer will be two. If you have selected two, if you had said two, congratulations, you know what DNA is. You have grasped this idea of a pitch. You've grasped this idea of a pitch. Pitch is one complete turn of DNA is a pitch. So one pitch second pitch. See, so what is a pitch? One complete turn of a DNA double helix. Double helix, you have to mention double helix, of course, or else you can also mention DNA like you can show DNA like this. Okay, double helix refers to this part. Okay, so this is pitch number one, this is pitch number two. I hope I'm making myself clear. This is my first pitch, this is my second pitch. So two is your answer. One complete turn. From here it's starting like this, for example like this. From here it's starting, it's going from here. It has started, it is shifting, over here it's like this, then again shifting, it's here. From here same again, it's shifting, and it's up over here, shifting up, again shifting over here straight. See, one complete pitch. Okay, have I made myself clear? Then focusing on the following parts. Okay, focusing on the following parts. This pitch, one single pitch is purposely I'm writing that in black. Purposely I'm writing that in black. Is 0.34 nanometer in length. Very small, very small. How much small? You can always guess it through this. 0.34 nanometer is 0.34 into 10 to the power minus 9 meters. So you have to multiply, you have to divide this by 10 to the power 9 to get the meter reading. So it's pretty small. Okay, okay, you have to like add 9 zeros after the point and then you get 34, which is like 0 0.12345678934 meters. See, so smaller value, but consists of 10 base pairs. Base pairs, what are the base pairs? The nitrogenous bases are pairing with each other. Adenine, guanine, adenine, thymine, cytosine, guanine, adenine, uracil. Okay, they're pairing with each other, base pairs. They're the base pairs. Codons and base pairs are the same thing. I hope everyone has understood this. I hope everyone has grasped this part. Okay, okay. This is how the DNA is pitched. Moving on to the next part. Okay, guys. One more small mistake I had made over here. I was checking the board. I had made a small mistake. Is that I had confused the numbers over here. My mistake. I'm sorry for that. I'm literally sorry for that. Oh, come on, people are frying me in the comments right now. People are frying me down. Come on. What the hell was that? Huh? Like you wrote a random number and you just wiped it off. What are we are a joke to you? Are we a joke to you? No, you aren't. You aren't a joke to me, my dear. My dear students, you aren't a joke to me. You people, I've taken you people very seriously, and hence we are having this lecture right now. One single pitch of a DNA is 3.4 nanometer in length, not 0.34. 0.34 is something different. 3.4 nanometer in length. Hence, it is 3.4 into 10 to the power minus 9 meters, which is again equal to 0.1234567 834 meters. This is the difference. Still small, but the number was a bit different. Now we have noted it down. In case you want to note it down, please do note it. Okay, you can pause the video and note it down. I'll be erasing the board. The base pairs. Achha, let me erase it first before I speak of it. The base pairs. Okay, so by now we know what is a pitch. What is a pitch of a DNA? Okay, so when the teacher asks you what is a pitch of a DNA, pitch of a DNA, like what is a pitch of a DNA? You say this: a complete turn of a DNA double helix. Okay, is a pitch of a DNA. You know what to do. You know what to write in your exams. I hope so. I hope so. You know, you people are studying. I know that. You people are studying. You people are serious in your studies. Of course, you people are serious in your studies. Don't worry. Now, there are base pairings over here. DNA bases. DNA bases. Okay, DNA bases over here. I won't be drawing it over here. It's going to be a pretty lengthy one. Same thing. DNA bases. Bases. Having hydrogen bonds. Having hydrogen bonds. Okay, having hydrogen bonds with them. Now, what is the distance between these two? First, for example, this is first number one base pair. This is my number two base pair. What is the distance? What is the distance between first base pair over here? First one, the first one, first base pair, and distance with the second base pair. What is the distance? What is the distance? Anyone? Since one pitch is 3.4 nanometer, if you remember, of course you do, and there are 10 base pairs situated in one pitch. One pitch is 3.4 nanometer in length. This is whole thing is 3.1 nanometer in length and consists of 10 base pairs. So if we divide this number by 10, we would get we would get 0.34 nanometer, right? We would get 0.34 nanometer. Now this is this. So now we know the distance between two base pairs can be summed up by dividing the total length of the pitch by the number of base pairs it houses. So 3.4 is the length, 3.4 nanometer is the length of the pitch, and the number of base pairs are 10. So we divided it by 10, divide the length by the number of base pairs situated, and we got 0.34 nanometer, which serves as the distance between the first and the second base pairs. Okay, now moving on to the last part. Of course, the first part which was present, that you always know that sugar and phosphate are the backbone. Sugar and phosphate always form the backbone. They are like the vertebral column of the DNA, not us. They are a genetic material. Without them, we wouldn't be surviving right now. We would have so much of information, genetic information, as in, okay, not any more information. Information stored hub is your brain, not your genetic material. The information which you gain throughout your life. Okay, not evolutionary information I'm talking about. I'm not talking about evolutionary information. I'm talking about the information gained throughout your life. That it's stored in your brain. The genetic material stores evolutionary information. Okay, evolutionary information as in when you adapt, and that adaptation has to continue all throughout your life. It's then when the DNA stores the information, the genetic material stores the information. Okay, got confused now? That thing will be cleared when I start evolution. Okay, so don't worry, the videos will flow in gradually. Of course, if you like this content, you would get quality and quantity over here. Okay, you will be getting extra information, some details, some random current affairs stuff. Okay, so don't worry. You can always revert to this channel for whatever information you want. You can comment down your questions. I will answer them personally. Personally, in the sense, I'll show it over here in the video and I'll answer those questions. Of course, I'll make a separate video dedicated to only questions coming from my audience, my beloved students. Okay, so now the voices should go unheard. Actually, won't go unheard. I would make sure. I will make sure. Okay, now this thing. The last part, if you remember from the thing I had shown previously, if you have had taken a screenshot, if you have written it down, whatever you have done, if you check that out, the last one was that this is a this is a sugar phosphate backbone. I'm drawing the linear structure of DNA. This is one nitrogenous base, another one nitrogenous base, and this is my another sugar phosphate backbone. I'm not drawing the primes anymore, you know it. This is another one. What is the distance between these two? What is the distance between these two? What is the distance? What is the distance? Tell me, tell me, tell me, tell me. Yeah, 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 please. Comment it down. Yeah, correct. This is 0.34 nanometer, which is also equal to 3 0.34 into 10 to the power minus 9 meters. Okay, this was the previous part. Now this is when I just zoomed into a part. Now we draw the bigger portion of the image. The base pairs are like this, very close to each other, very close to each other. See the base pairs very close to each other, very close. I'm just zooming in this part. Okay, I'm just zooming in this part. These two, the pi electrons of the above base pair will interfere with the pi electrons of the base pair below it. Number one. And in presence of hydrogen, in presence of hydrogen, the interaction would be more efficient, would be more effective. Okay, the reaction would be more vigorous, more effective, and there will be something known as stacking. The pi electrons of the base pair above will interact pi electron will interact with the pi electrons of base pair below, and in the pres in the also in the presence of hydrogen. Where is the hydrogen present? In the bonds. In the presence of hydrogen, the effectiveness or the effect increases. Okay, the effect increases. What happens when the effect increases? This effect will give the lower base pair more stability. This effect increases the stability. This effect, stacking will increase the stability. Why? Because this will interfere with its pi electrons. The pi electrons will interfere somewhere over here. We know it through the diagram. Chemistry will teach us. We know it. The pi electrons will interact with each other, and hence they will be bonded somewhere over here, bonded to each other. If you remember this, they will be bonded to each other through electronic orbitals, if you remember the concept of orbitals. See, speaking of chemistry and physics in bio, of course, three daughters of science goes hand in hand. The pi electrons will bind to each other, will also use hydrogen and make it more effective. And hence, if this is connected to this won't let this one move out, right? This won't let this one move out. Hence, this and this will be in contact with each other. Okay, if this is one of my nitrogenous base pair, this is one of them, and these are my pi electrons, and they get intermingled like this. If this even wants to leave, these will not let go. These will not let go. So each and every base pair above will have a stacking effect on each and every base pair below. Okay, and that's to give more stability to it. And in presence of hydrogen, the effect increases, the efficiency increases, and hence the effect of stability. Okay, now the DNA is quite long. DNA is quite long. DNA is quite long. How much long? Quite long. We have heard it 3 times from you now. How much long? How much long? Let's see. Let's see how much long. No, I'll just erase the board. So guys, the length of the DNA, the length of the DNA will be. Let's not write it down. Let's find it out. How do we find the length? What do? We take the total number of base pairs present. The total number of base pairs present in a human DNA is this 6.6 into 10 to the power 9 base pairs. And now if we multiply it with a with the distance in between each and every base pair, of course this is in meters. We have to find it out. If we want to find it in meters, of course, why not? If we are getting the chance, we'll cancel this one out. These two are gonna cancel out. So 6.6 into 0.34 will approximately give you 2.2 meters. Meters. This is a base pair. So 2.2 meters. DNA, each and every DNA strand is 2 point is roughly, of course, roughly 2.2 meters in length. Each and every DNA strand is roughly 2.2 meters in length. Roughly. You can always do the calculation. The calculation would come up to calculation is 6.6 into 0.34. It's 6.6, 0.3. Yeah, calculations will come up to this 2.244. Since in maths we know if we have 4, we cannot convert this number to the higher decimal value, to the higher value. So we just consider this. Let's consider this. This one is a more smaller part. Okay, even if we leave it, nothing would happen. Nothing would go wrong as such. So this is approximately 2.2 meters. Okay, now each and every DNA molecule, each and every DNA molecule, DNA is a molecule, right? Each and every DNA molecule is, if we open each and every strand, is 2.2 meters in length. Each and every strand, so there are millions of DNA strands. It's roughly so long that the nucleus is a small thing, and the DNA is 2.2 meters, more than the length of this board. More than the length of this board, because this is 60 centimeter. 2.2 meters is more than length, and this nucleus is not even this size. Okay, not even this size, not even this. It's small. It's smaller than a dot. It's smaller than a dot. How will it accommodate this like so huge a structure? Let's check it out. So it needs to be packaged. Okay, some big stuffs we have for gifts to give it to someone. Gifts for special people. Special people. See. I can see the smile. I can feel you. Okay, I can feel each and every student right here, right now. The smile says it all. Okay, for example, you're going to gift something to someone very special. Someone is special to you. Can be anyone. Can be anyone, of course. Someone is special to you, and your gift appears to be more in size than the box you brought it to pack it for. The box you brought it to pack it in. The gift appears to be more in size than the box. Hence, what you will do? You will push the gift inside. You will just attach. You just put the box. You pack it properly so that the gift doesn't just bulge out of the box. Okay, you won't just put it in the box and just close it half out. it's half out of the box and just go and give it. No. uh what is it? It's the impression that matters, it's presentation that matters. Of course presentation matters, yeah. So what you will do, you will just, you might as well push the gift inside, you must just push the gift inside a bit, it will make it, force it to accommodate, the box might bulge out, no problem. We'll just pack it, we'll pack it with a nice paper and make sure that the box looks nice, right? Same thing goes with RNA, not RNA, DNA. Say same thing goes with the accommodation. What does happen? What does happen? We know there's DNA. DNA is slightly negatively charged. DNA is negatively charged. So we'll take a protein. I'll tell you the name a bit later. We'll take a protein that will be positive in charge. Positive charge. Why protein? Why not any other DNA? Because when we mix genetic material, it's going to create a new, it's going to create a new being, and not a being, it would create a new genetic material. Two DNAs fused together will create a new DNA. It will create a new DNA. So not always does it create a new DNA. You always require some stuffs. Okay, not it's like put one DNA, another DNA, and then boom, a new DNA is created. No, you need various stuffs like restriction enzymes, DNA ligase, transcriptase, everything. Not transcriptase as such, DNA ligase, DNA polymerase, polymerase 1, 2, 3, whatever you have, polymerase. Okay, and we'll be coming in those parts later on when we do RDT, recombinant DNA technology. That's a pretty important part. Biotech is the most important chapter you can ever think of in in biology. Okay, DNA, I'll take a protein, positively charged. This protein is known as his stone, his stones. Okay, not his stones. No, we never say that. We never say that. This is his stones. One single term, his stones. Okay, histones. Now these histones are of five types. Five types. Okay. So histones are of five types. Not actually five, I would say. Four. Five types. Five types. Five types. Not five. Not actually five. I would like. Sorry for saying that. It's five types. DNA is of five types. H1, H2. H2 has two types in itself. H2A, H2B, H3, and H4. Okay, now these all come together and pack the DNA. What do they do? What do they do? They form an octamer. They form an octamer. Okay, they form an octamer over here. I'm drawing a 3D structure of the DNA octamer. I won't be able to draw this because it's a bit lenient like this. It's going like this. There is one more behind this. Okay, if I'm drawing it like this, it would be somewhere like this. One more thing, if I drew it like this part would get hidden and this part. Okay, there are four parts to DNA octamers. Okay, if I'm drawing it in the pie format, if I'm just separating it out, this is one part. Let's draw it like this. This is one part. This is one part. One part. Okay, one part. First part. Second part. Second part. Then over here, third part. Over here, I'll be drawing it like this. Fourth part. Okay, over here. One, two, and it's divided actually over here. It's divided. There are eight parts to it. There are eight parts to it. There are eight parts to it. Same face over here. You can see it like this. You can see it over there. Okay, this is a 3D structure. It's kind of a 3D structure. You can not see the other side, of course. You cannot see the other side. One, two, three, four, one whole thing, two, three, and the fourth one you can't see. It's somewhere behind. Somewhere behind. It's like this. It's somewhere behind. It's somewhere behind. Okay, somewhere behind. Somewhere behind, which cannot be seen right now. This is one octomer. See, one, two, three, four, five, six, seven, and over there, this is the fourth one. Over there is the eighth one. Okay, behind this board is the eighth one, which you cannot see right now. Which you cannot see. It's in the z-axis, to be sure. It's a negative z-axis. Remember this axis in physics? Everyone remembers this. So negative z-axis. It's in this axis actually. Negative z-axis. It's behind. It's behind. Okay. These are in y and x axis. These can still be seen. They are in the negative z-axis, but bit tilted on the y-axis. But this one cannot be seen. It's direct parallel to the z-axis, negative z-axis. Okay. So these DNA are like this: H2B, H2A, H3, and H4. Same way over there: H2B, H2A. Oh, H2A will be down. Sorry, my mistake. H2A would be down. This is H3. H3. H2A is over there, which I was explaining previously, and this is H4. This is H4. They are octamers. Okay, they are the octamer. H1 will come a bit later on. So if we consider this as one circle, one protein, DNA, what do they do? This protein is over here. DNA will come and wrap itself around this protein. Wrap itself around in the protein. On purpose. Okay, I'm just trying the ones going from the back. It will be dotted because it's going from the back. Okay, so it's what is it doing? It's wrapping around because DNA is negatively charged. It will be attracted from the positively charged. So this is positive. DNA is negative, so DNA will wrap around this. DNA will wrap around this means that it has shortened up. Now these two faces will move out, will project out, and it's then the H1 comes in and clips it. Okay, it will clip it. Which one will come and clip the parts? So we can see that DNA is now clipped. This part is clipped, held. It won't open easily. You need enzymes to open it, of course. It won't open it easily. This whole thing is known as a nucleosome. Okay, this nucleosome consists of 200 base pairs each. Over here, one nucleosome consists of 200 base pairs each. Now, let's see how many base pairs will curl up and how many base pairs will form over here. I'm erasing this part. So, around the protein, we know 200 is there. Around the protein, you will find 147 base pairs of the core DNA. The DNA that curls around the nucleosome is known as the core DNA. DNA curling around, okay. DNA that curls around the histone, histone octamer is known as the core DNA. Okay. Then from 200, if we remove 147 from 200, if we remove 147, we are left with we are left with 53 base pairs. Now, this 53 base pairs from the linker DNA. What DNA? Linker DNA. Linker. Why linker? See, it's getting linked over here. So if we divide it in this part, this is my this is my core DNA. Core DNA consists of 147 base pair, right? Consisting of 147 base pairs. Clear? And this part is my linker DNA. Linker DNA consisting of 53 base pairs. Okay, so this is the whole thing. So this one is my H1 histone, the histone which is clipping the DNA together. Of course, if you take a ball and wind a string around it and take two ends. Okay, take two ends. After some time, you might see anyone tugs at one end and the ball will rotate and the strings will come out. Strings will come out, will unwind. For that not to happen, okay, if that happens, the DNA will again go back to its normal shape. Of course, have you tried uh what do you say, have you tried winding a string up a ball? Always try that and you can always have your results. Okay, you can see that the string will not stay on the ball will start rotating and your string will unwind at one of the ends. So over here, if you wind it and then hold two of the strings, if you wind the whole thing and hold both of the ends of the string together in your hand, even if the ball moves, the string won't unwind, because string can't, there won't be any source. Same thing is happening. The ball is over here, the DNA is winding, DNA is a string that is winding, and each one is coming and pressing it down, holding it down. So there are so that's how the DNA is packed so that it doesn't open. Of course, you have the remote in your hand, remote or keyboard or the screen, the phone in your hand. You can always stop the video, pause the lesson, and you can copy it down in case you have missed it. You can again rewind and replay. Okay, now that's what happens. The DNA molecules now, each and every DNA is, this is one histone octamer. Here's a DNA that's going linked with one each one. Again, it's going up. Take a ball is over here. Again, it's linked. Again, it's going up. One ball is over here. Again, this part it's going like this actually. And it's getting linked. Okay, the linker DNA is out here, is linking the part. So on and so forth. So coiling is happening. See how much of DNA molecules are going in, how many DNA molecules are being struck, how many DNA molecules are being stuck against, how many DNA molecules are being stacked, not in that sense which I just explained, not that stacking. This is hoarding, like economic terms. Hoarding up means having a collection of something in your warehouse. Okay, something in your warehouse, you collect something. This is a collection of the DNA. Now the DNA strand, this strand was pretty long, this just came around, wound around the ball, the histone octamer, and the size shortened. So the open DNA present in a helix form. Of course, DNA is present in a helix form. Starts curling around this. Okay, I'm just showing it in the form of one single line. Okay, one single line. This is being represented as one single line. Okay, representation purpose. So it's coming, it's curling, and coming out. Linker again coming, curling and coming out. Linker over here, ball over here, octamer over here, linker over here, octamer over here. That's how the DNA is getting packed. This is this is known as condensation. So when, if anyone says DNA condenses, the chromatin network condenses to form chromosomes, this is what they mean. It's condensing, and then these again come close together. These come close together, causing supercoiling. Supercoiling. This supercoiling, this supercoiling will give rise to our very own chromosome. This part is centromere. Yeah. Let that be alone. Vary on chromosome, right? A vary on chromosome. This is the part which is geneless chromosome. Okay. So guys, now focusing on after this supercoiling occurs, we have the condensed form of our what do you could say chromosome. This is something different. We are going to discuss this in the molecular basis of inheritance part, not as such. Of course, we will be doing that when we are focusing on Gregor Johannes Mendel, Sir Mendel. He did a lot of like father of genetics. He is. Of course, we all know that. Being bio students, we always know that. There are some basic terms that needs to be brushed upon, but nothing else. Nothing else. So now this chromatin, you now this chromosome. Some places, the balls or the what is it, histone octamers are situated close to each other. Okay, pretty close to each other. So each and every DNA molecule winding up will be pretty close to each other. Okay, it's gonna be pretty close to each other. See, so close they are to each other. So close they are to each other. Now we can as well say that they are densely packed. These guys are densely packed. Densely packed. We can also see another condition whether there's one more ball over here which gets its packaging. Not ball actually DNA. This heterochromatic and sorry for the term. This is the nucleosome, and then you get somewhere over here, then you get it somewhere over here. The ball is a greater one. Not ball, histone octamer. What has happened now? Okay, these are link link from H1. Again, H1 is linking, H1 is linking, H1 is linking. Okay, see, these are less densely or rarely packed. Rarely packed. We know in a nucleus, we can see a nucleolus. That's out of our topic right now. And we can see various net-like structures. We can see various net-like structures like this. These are the chromatin. These are the chromatin. Now they are actually like this. They look like this. With the histones backing them. Histones backing, histone backing, histones packaging them. Okay, histones packaging. Okay. Now see, this is densely packed and known as heterochromatin. This is rarely packed and known as euchromatin. Okay, densely packed hetero, rarely packed euchromatin. At some parts of the nucleus. Let's draw this in blue. This will be a better idea. Okay, the light parts could be always explained. This is a nucleus. This is a nucleolus, and we can see various parts, light colored parts. Of course, we can see light colored parts. We can also see that some parts are darkened. Some parts are darkened. Some Parts are again darkened. Dark parts. The ones in black are the heterochromatin. Heterochromatin is in black, and euchromatin is in blue. Now, what's the difference? What is the difference? Let's see. Let's check it out. Heterochromatin. Heterochromatin is transcriptionally. Now, what is transcription? I'll come to it a bit later. Transcriptionally transcriptionally inactive, whereas euchromatin is transcriptionally active. Okay, okay. I'll be doing what is transcription when we do Central Dogma. I focused on this one before we do Central Dogma because, of course, that is something which you will understand after this. Okay, we need to know how the DNA is packaged. Transcriptionally inactive, transcriptionally active. Why? Because this is densely packed. This is densely packed. This is densely packed, and 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 there won't for transcription to occur. Transcription is a process. For now, just know transcription is a process. For transcription to occur, we need an enzyme. We need an enzyme known as transcription RNA polymerase. RNA poly. Okay, you can always say RNA poly also. Whatever you say. And this one does nothing but just sits on the DNA, uncoils it, and starts reading. Now, do you think anything would be able to pass from these lanes? Because I don't think so. I don't think so. I don't think so that anything would be able to pass from here. Anything would be able to pass. But over here, see, there's a very big gap. There's a big gap over here in between them. In between them, not only in this part, only in this part also you can see there isn't any space. There won't be anything. Once a RNA is polymer sitting over is going, it might hinder it. It might hinder. It might get a hindrance from another strand. It might change. Suddenly it's going, and suddenly it's changing. Now, what will happen if it changes its route? Coming to it a bit later. See, if this RNA poly is sitting over here and transcriptioning the whole part, it won't come across any hindrance and would again go to the next part properly. Over here, if it's going like this, it's gonna come across any of the hindrances and it might as well change its course, change its route. So now what is transcription? This urge to know. That's the thing I want. That's the thing I want. I want this hunger to grow more. This is densely packed known as heterochromatin, which is darker areas. Darker areas in a nucleus of a cell are known as heterochromatin. They are transcriptionally inactive. On the other hand, there are some light colored parts of a DNA. Of course, once you stain them, always have to a you have to always stain the nucleus to take a look at its chromatin material. You stained it, you got this, some dark, some light bands are the euchromatin, because see, so much of gap. Okay, if I just concentrate black color over here, see, so much, it's so dark because it's concentrated, it's concentrated, see, it's dark. But same way, if I put it like this, with proper spacing, you would easily be able to recognize. See, oh, it's looking gingy, and you won't be able to like recognize it. This is a deeper shade of black, this is a lighter shade of black. You can easily understand because it has 5 to 6 coatings, and then this one has only one, so you would be able to differentiate. Of course, same dye when present in concentrated, when present in concentration, see, each and every part is taking up the dye. They are in concentration, they are densely packed. The concentration is high over here, hence the dye is showing deeper bands over here until unless the concentration is over. And in the lesser concentrated part, you can always see the light color because the dye is evenly spread out. Each and every part has taken exact amount of dye, same amount of dye, equal amount of dye, and each and every part is glowing different. Over here, each and every part is glowing different, but since they are close to each other, the color is appearing to be one whole color. Okay, this is clear. Now I'll move into the central dogma, and then we'll end the video. Okay. Then we'll end the video. This video is going to be a bit long because I discussed nearly each and every aspect of the DNA which you need to know for your further studies, for the dive in biotech. Okay, because you will require that in biotech, and biotech is not easy until and unless you have understood the stuff. Now when we are focusing on central dogma, it is a cell made protocol. Cell made protocol. What are protocols? Protocols are set of rules. Okay, protocols are a set of rules. What are protocols? Protocols are set of rules. Okay, for doing something. So cell made protocol means cell made set of rules. What are the rules? A DNA can give rise to DNA. This process is known as replication. That same DNA can again give rise to an RNA. This process is known as transcription. Transcription. Let's draw it a bit bigger. I'm erasing this one out. DNA giving rise to RNA through transcription. Transcription. Remember the term transcription. RNA giving rise to protein through translation. Translation. This is the whole central dogma. DNA can replicate itself. DNA can replicate itself. Now DNA through the process of replication is creating another DNA. DNA through the process of transcription is creating an RNA. RNA through the process of translation is creating a protein. Now this process is reversible. It can happen both ways. Reversible means it can happen both ways. This is known as reverse transcription. Reverse transcription. Okay, that process is known as reverse transcription. This is the central dogma, cell made protocol, a cell made set of rules. You can copy it down if you don't know it. If you know it, well and good. Okay,